textbook. But let's talk about the nucleus, because this is the brain, this is the command center, this is the library of your cells. And why do I say that? Well, the nucleus is where you find nucleic acids. More specifically, that's where you find the majority of the DNA in your cell. So again, nucleic acids, these are long polymers, these chains of nucleotides, and DNA, these have deoxyribonucleic acids, and RNA it stands for ribonucleic acid. Now, this is the structure of DNA, that double helix. And what does it mean by that? Well, you have two strands of DNA, and they're kind of twisted around. Again, that helical shape referring to some sort of spiral. And what we have with DNA, well, your DNA, you have actually 3 billion base pairs. So if you see in this previous picture, each of these pairings is one base pair. So you actually have in each of your nuclei about 3 billion of these pairings in just one nucleus. So your DNA has that many, and if you took all the DNA, all the chromosomes inside of just one nucleus and stretched it out end to end, it would be about 6 feet long end to end. So again, this is the nucleus, has a lot of the majority of your chromosome, or has your chromosomal DNA. Not talking about mitochondrial DNA, but just nuclear DNA. So if you took all the DNA in the nucleus, it would be about as tall as this 6-foot guy right here. So a cell nucleus is about 10 micrometers across, so that's very, very tiny. So how tiny is a micrometer? Well, if you have a human hair, somehow a cell nucleus, is actually super tiny. Like if we did this to scale, it would be very, 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 very small. You can't even see it in here. And then, so how do you fit all of this DNA, which would be 6 foot long, into something you can't even see? And so how do you do that? Well, this is what we have here. So DNA packaging. Look at her long hair, and how can she make it into more compact air, or how do you take long hair and make it into a more compact structure? You wind it up and over, up and up until it's kind of this tight bun, right? Or it's not just women who can do that, men can do that as well. But this is how your DNA is able to take 6 feet of DNA and compact it into something your eye can't see. So what you have is DNA, and it's wrapping around these little globs here are little proteins called histones. And that's a little, like maybe if you're taking cell biology, or if you're a bio major, or if you're going to, well, maybe nurses need to know this too. But what happens is that these proteins help to wind up the DNA, and then if you wind up and coil this DNA over and over again, you get these structures called chromosomes. So again, it's like trying to take a long, very thin structure, but making it more compact and easier to work with. So this is what we have here, and again, DNA is very long, but you're able to compact it. Or if you remember Moana, what did she do one time, like to get her hair out of the way? She has that long flowing hair, and remember she took it, she wrapped it around and made that tita bun, and it's very compact, right? Very neat, instead of very long and flowing. Same with DNA. Your cells are able to kind of unfurl their DNA and also compact them when needed. Now, nucleotides, let's talk about this again. So remember that a basic nucleotide has a phosphate, a sugar, and a base. And this part right here, the base part is the one that can vary. Like remember those 4 letters in DNA, AU, or not A, ATCG. But this is DNA and RNA. So what we have here, ribose is a sugar. It is a monosaccharide. Here's other examples of monosaccharides, but it doesn't have 6 carbons. It's 5 carbons. So that's what we have here. So ribose, oh yeah, sometimes it's been doing that with the live lectures. Sometimes it doesn't publish as public by default. It goes by unlisted. Don't worry, I'll handle that after this lecture. But again, back to this. So thank you for that. Well, I can't say names during broadcast because IT said don't do that. But anyway, back to ribose. So ribose is a sugar, but what's deoxyribose? What's the difference? Well, the thing is that this is an oxygen or a hydroxyl group actually. But the difference is that it's pretty much one oxygen atom. So deoxyribose has one less oxygen. So whenever you see that prefix D, it's like when you have contaminations versus decontamination. Decontamination, you're removing the contamination. Deoxyribose is ribose, but you're minus 1 oxygen. So that's the difference between deoxyribose versus ribose. But the overall structure is very similar. But this is what the difference is between DNA and RNA, just one less oxygen molecule on that sugar. So again, DNA has this set of 4 nucleobases. So you have the sugar and phosphates, but it can vary which nucleobase you have along a nucleic acid. Whereas RNA, you have these 4, and again, the RNA does not have thymine, it has uracil instead. And this is how you link the nucleic acid. So remember our previous example, it's kind of like that friendship bracelet. You have that constant part that repeats over and over again, but it's these nucleobases that dangle off. This varies depending on the sequence and chromosome and gene. And we'll get into chromosomes and gene real quick. So why is it important to know these bases? Well, it's a phenomenon called base pairing. So the thing about these bases, they don't randomly link together. And it's not always like, it's not going to put an A linked to an A or a G linked to a T. There's always a consistent basing, or I should say like there should be a consistent basing, because this is how, when it doesn't match up correctly, this is what causes problems with your cells and your DNA. But anyway, so the normal pairing for these nucleic bases are A and T and C and G. So, but the thing is that with RNA, remember RNA doesn't have thymine, it has uracil instead. So instead of A and T, and if you have DNA binding to RNA or RNA binding to RNA, it's A to U. Okay, so here's a nice mnemonic I like. So a teacher asked you to get coffee. And I know this is not the grammatical U, but why does this work? Well, it tells you the base pairings. So you have A and T, G and C in DNA. But again, in RNA, there's that little twist. So this is why it's a U here. So instead of T, you have a A and U. So A binds to T, A binds to U, G binds to C. Now, the thing is that why is that important? Why do we care about base pairing? Is that nice to know or need to know? Well, the thing is that nucleic acids, these contain information. So DNA has information on how to make every single protein in your body, how to make different types of cells, how to respond to hormones, and pretty much anything or everything your cells can potentially do. Now, your cells don't do everything, like every cell in your body doesn't do the job of every other cell. Like your neurons aren't going to work like your muscle cells aren't going to work like your blood cells. But in theory, like when you start out as an embryo and then you start developing these different types of cells, all of that information to make those different types of cells is encoded in your DNA. So this is why nucleic acids are very important. Now, the thing is that what do you do with all the information in nucleic acids? One major function is to make proteins. So how do you make a protein? It involves DNA. So from a DNA, so I like to think of the DNA as your permanent record, because the thing is that what if you start changing your DNA? Well, if it's changing your DNA, then it's also going to affect how you make proteins. So from DNA to protein, you actually have an intermediate step. You have to make messenger RNA, and this is through a process called transcription and RNA polymerase. Hey, you see that last 3 letters of RNA polymerase? ASE. That means it's probably an enzyme, and it is an enzyme. It's an enzyme that makes RNA from DNA. But so mRNA is kind of like, I like to think of it as working copies. Like DNA is the stuff that you want to be the same in all of your cells, or most of your cells. We talk about immunology later on, or if you want to know more about the special stuff, you want to go, you might want to take that 400 level class later on in your senior year. But so sidetrack there. So mRNA, this DNA, you want that to be consistent, because if you change your DNA, you'll change your mRNA. And from the mRNA, this is where you make the protein. So you have the information to make every single protein in your body encoded in your DNA. But if you start altering your DNA, you have the potential to alter the proteins and how much of these proteins you make. So when you go from RNA to mRNA to protein, it's a different process, so it's not transcription again. It's something you call translation. So I know it sounds very similar, transcription and translation, but they refer to 2 separate processes. So again, from DNA to RNA, you have transcription. From RNA to protein, you have translation. And so this ribosome. This ribosome is an enzyme. So again, it's an exception to the ASE rule. So it doesn't end with ACE, but it's still an enzyme. And actually, ribosomes are also an exception to that rule or general rule that most enzymes are proteins. Ribosomes are actually kind of cool. They're actually a mixture of RNA and proteins. yet they still act as an enzyme. So this is one example of exceptions to rules of enzymes. Okay, so nuclear DNA, I remember this is like you have all this information, you have 6 feet of DNA somehow crammed into your nucleus, and this is why I like to refer to it like a permanent record. So this is, if you're on campus, you might be familiar with Hamilton Library. You can check out books, but there are some books that are so precious and valuable, you can't actually take them out of the library. You can open them, you can get the information from them, but they can't actually exit the library. So those type of books, those ones you can't change, you can't take out or check out, they're kind of like the DNA in your nucleus. So you can get the information from there, but this DNA should not be altered and it should not leave the nucleus, just like those special books in the library. Now, there are different types of RNA. So the one we talked about when you go from DNA to RNA via transcription, that's what we call mRNA. That's the product of taking the information from DNA and encoding it into mRNA. And this is the one that you eventually make into a protein via translation. Now, transfer RNA, they're a different type. They don't encode proteins. They're important in shuttling amino acids to a ribosome. And ribosomal RNA, so this is rRNA, it's not translated into a protein, but it's actually that part of a ribosome that's not protein. So it's interesting. Ribosomes have a complex molecular structure where it's like RNA and proteins all kind of mashed together, and somehow that works as a really cool enzyme. So what I want you to know for this at this level is that these two help with protein synthesis, but it's the mRNA that actually is made into a protein or encodes a protein. So again, how do you go from DNA to mRNA? So that DNA, that double helix structure, so you can actually, there's a special enzyme, again, that RNA polymerase. Actually other enzymes help to unwind it. They don't show it in this part, but what you're seeing is that RNA polymerase is kind of like reading the DNA, and then it's making a new mRNA transcript. And this is how it is. So do I want you to know all these steps? I think that's more appropriate for the 200 level biology level. But I definitely want you to know which enzyme is able to make RNA from the information in DNA. And the individual steps, that's, again, don't go too much into weeds. I think it's also it might be in the OpenStax book as well. But what I want you to know is like, okay, how do you go from DNA to mRNA? It involves RNA polymerase. And what you see is that it's using base pairing to synthesize this new mRNA molecule. Okay. So I like to think of it this way. Just like how my analogy, like DNA are like books in the library you can't check out, RNA polymerase somehow gets the information from that permanent record. So RNA polymerase is like the photocopiers, or if you print out a document from the library. You still have the information, but now it's something you can take out of the library and then do whatever you want with it. You can cut it up, you can photocopy it more, you can follow the instructions or learn from that. RNA polymerase is like those photocopiers. It takes the information from that permanent record and gives you working copies you can take out of that depository and work with. Okay, so this part is called transcription, so it's like RNA polymerase, this analogy. So again, this book, you're taking the information, but you're now making a working copy, and this primary transcript is your messenger RNA. So transcription makes an mRNA copy of DNA. So this is the process. But so the thing is that the mRNA still has the information from the DNA, but now you can take it out of the nucleus and you can actually alter it. You can actually change like what we call splicing. Do I want you to know too much about splicing at this level? Again, this is something I think is more appropriate for advanced biology. And at this level, you should just know how information flows from DNA to mRNA. So how do you encode that information? Well, this is where we have to talk about that base pairing. So here is an example sequence of DNA. So here I'm showing DNA with the blue sugars over here, and here's the mRNA molecule. And how is it going to pair? And so how is RNA polymerase going to make that new mRNA molecule? It's going to use DNA nucleic acid base pairing to match it accordingly. And again, these are the correct base pairing between DNA and RNA. Now, why is that important? Well, say you send off that mRNA molecule, and every time you have RNA polymerase coming to the same DNA molecule, it's always going to generate the same sequence. So you have this consistency due to base pairing. It's not just going to put random nucleotides in a sequence. It has to match that base pairing to generate a consistent match and consistent mRNA molecule every time from the same original DNA template. Do you not need to know the different stages? Know the stages as much as I'm covering here. If you're talking about initiation, elongation, and all those other things, I think that's something for more advanced A&P and also molecular biology. I'm more concerned about how information flows from DNA to a protein. Now, translation. So what we just covered with going from DNA to mRNA is transcription. Translation is different. So what you do for translation is you take RNA and then you finally make that protein. And again, those ribosomes, those enzymes, very important. So what you're doing is creating polypeptides from mRNA. So in this cartoon, each of these little beads is amino acid. Remember, those amino acids are linked via peptide bonds. So when we have many peptide bonds, you have a polypeptide. So the thing is that ribosomes, these are enzymes. Just like RNA polymerase reads DNA to make mRNA, ribosomes read mRNA and translate it into proteins. So what we have here is a ribosome. So what do they do? So they take all these amino acids, and again, at this level, you should just know that there are 20 general amino acids, not including selenocysteine. So amino acids are taken, and actually this is where tRNA comes in handy. So tRNA is actually responsible for ferrying amino acids to a ribosome, and then the ribosome reads a mRNA molecule, makes a polypeptide by putting the linked amino acids together via peptide bonds, and then you finally get a polypeptide. So the overall process is called translation. Now, this is where I don't want you to get too much into weeds. This is just for your information, but this is how you're able to actually get mRNA to be from mRNA to make a protein. So this is what we have here. So this blue blob is a ribosome, and this little, how would I describe this? This kind of I've heard described like kind of like a cross or a little blob right here, but this is a tRNA, and this is actually amino acid. So notice that these tRNAs are actually carrying the amino acids, and then this ribosome is starting to connect all the amino acids together. That's pretty much what I want you to know. Do I want you to know all this information over down here? This is more FYI. I just want you to know that ribosomes are the ones that read mRNA and they make a polypeptide from it. That's pretty much it. Now the genetic code, you might have seen that, but what does this alphabet soup mean? Well, notice that you see that there are these 4 letters, and DNA you have the 4 letters. So A, T, C, G, and mRNA, instead of T, you have U. But notice that when you go from this here to over here, you notice that you have this kind of base pairing, right? So A and T, A and U. So these follow these base pairings. You just flip it around. And then what you have is that the interesting thing is that how does the ribosome know which amino acid to put at what position? Well, they actually read 3 base pairs, or not base pairs. They read 3 bases of an mRNA molecule at a time, and then each possible combination of 3 bases corresponds to an amino acid. So again, genetic code. Do I want you to know and memorize what triplet means what amino acid? No, that is. I don't think even molecular biology text, because there are 64 potential codons, and I don't think they're not going to test you, or at least when I was an undergrad, they didn't test me on that. You're not going to be tested on that here. But what you should know, the genetic code refers to that whole sequence of these bases, all those letters. So the bases are kind of like the alphabet. So in DNA, you have a 4-letter alphabet. And RNA, you also have a 4-letter alphabet. Mostly the same, but you just change U and T. Yeah, so don't know about initiation, elongation, and termination. If you're studying that, that's beyond the scope of this class. All right, so codons, these are like the words of the genetic code. And the words are 3 bases, so it's consistent. Ribosomes read 3 bases at a time, so this doesn't really change. So when you have a gene, it's like putting multiple words together in a sentence. So genes are kind of like paragraphs or novels. So just like how you put words together to describe something or to give information on how to make something, same with genes. So by having all these codons together, they basically give you the information to make a new polypeptide. Now translation. So translation, this is where it's often kind of like easily confused. So translation, like say, or if you're talking about someone about translation in a non-molecular biology context, what do they think of? Well, they might think of translating between languages, right? So what we have here is a translation, same information, but now you're going to a different language. So yeah, I study anatomy and physiology. J'étudie l'anatomie et la physiologie in French. I know I need to do more in Duolingo. But same information, just a different language. So when you're going from mRNA, you're going to a different language with proteins. You still have that same sequence, but now you're translating to a different language. So this is another way I remember it between transcription and translation. With transcription, you're going from DNA, you're going from nucleic acid to nucleic acid. But when you're going from mRNA to protein, you're going from nucleic acids to amino acids. So again, DNA to RNA, nucleic acid to nucleic acid. mRNA to protein, you're going from nucleic acids to amino acids. So a different language altogether. At least that's how I like to think of it. So mRNA, so this is another analogy. mRNA is like a recipe, and the ribosome is the enzyme that takes the information from that recipe, and then it takes all the amino acids, and based on the information in the mRNA, it's going to assemble that protein based on that information. So you might have heard of this if you've taken a biology class, central dogma. So I know it's like kind of an outdated term, but when you're starting out, it helps to convey how information is transferred from DNA to make a protein. So it's like a recipe book, the very old recipe book you don't want to alter or damage even further. Then transcription is how you get that information from the recipe book. So when you're making an mRNA transcript, you're not copying the entire genome and all the 3 billion base pairs in a DNA molecule. Your cells are kind of choosing what information they need to make what proteins they need. So when you have the partial transcript of that original DNA, you have translated it, and then you finally get that polypeptide you can use to make the proteins in your body. And how is this very relevant? So this is how if you've had the Pfizer Moderna mRNA vaccines, this is the mRNA we were talking about is the same mRNA that is the core of these vaccines. Now here's that infamous coronavirus, but are they injecting the entire coronavirus into your system? They're actually not. The cool thing about this is that they actually only take the part that encodes those proteins. So they only take a tiny part of the sequence. They don't take the whole information of a virus. They only take the part that encodes these spike proteins. So spike proteins, what they do is they only take the small part, the one that has these spike proteins on the outside. And this is how they do the vaccination. They have all these, like, remember those phospholipid membranes, and so it's kind of like they have an artificial membrane. So what can this membrane do? It can actually help to deliver and carry these mRNA or mRNA molecules into your cells, because again, these mRNA molecules by themselves, they would actually have a hard time. So by making it kind of like a vesicle, it's almost like endocytosis, that moving of or shuttling from something from the inside, or actually not quite endocytosis because it's not actually pinching. I retract that part. But somehow this is how you're able to actually get that mRNA inside your cells. So what happens is that this mRNA goes to a ribosome. That ribosome translates it and makes a spike protein. And this spike protein is how it's processed by your cell even further, and then it presents it to immune cells. So this is how these vaccines work. And you might have heard, like, so again, translation or transcription to translation. So DNA to mRNA protein. The cool thing about the Pfizer and Moderna vaccines is that they kind of take a shortcut. They don't inject the entire viral genome because you don't want that virus to actually replicate in your body. They only take the part that encodes that spike protein, and then when you deliver that mRNA into your cells make the spike protein, and then they can present it to your immune system, so your body is able to recognize the spike proteins of a coronavirus before it's actually infected with a coronavirus. So again, common misconception is like they're injecting the entire virus with these vaccines. Nope, they're only using this part and the spike protein. The spike proteins are only on the outside. They don't actually contain all the enzymes and information the coronaviruses actually need to replicate. So it's like if you just took someone's hand and then just like tried to make a new person from that person's hand only. It doesn't have the reproductive organs, and that's the same thing with the spike protein RNA in these vaccines. They're only trying to use a part that your cells, your immune cells can recognize, so it can actually combat the full virus. And another thing you may have heard, misconception you may have heard, is like these mRNA vaccines are altering your DNA. If you look at the flow of information, mRNA doesn't flow back to DNA. mRNA does not alter DNA. So again, this is where a lot of misinformation is happening. That flows from RNA to protein. It doesn't flow the other way around. So Pfizer and Moderna do not alter your DNA. How would your immune cells know that spike protein belongs to a virus? So the thing is that your immune cells know when they're actually brought up, they know what's already part of your body. But if you all of a sudden introduce a new protein, they're like, hey, this wasn't in the bone marrow. This wasn't what we saw when we were initially made. This is probably a foreign particle. So you're not going to make spike proteins naturally. So their immune cells, especially in your adaptive immune system, they're really smart about recognizing things that are foreign to your body. And breakthrough cases are occurring the vaccination, and why many of those cases are less severe. So fully vaccinated, why those are happening fully vaccinated are, I think one of the theories is like maybe we have declining, like this is why we need booster shots sometimes for some other vaccines. Sometimes we have like our cells, our memory cells are left over from a vaccine, they might decrease over time. And this is why they have to, and what this vaccine has been out, maybe the people who got the initial trials, maybe less than a year, so we don't know how long this protection lasts. Yeah, and then, but yeah, immunology, trust me, we can get a lot into the weeds with the immunology. We don't have time. If you're really interested in it, my immunology lectures on my same YouTube channel, and there's also the stuff in the chapter, but it is a big topic. Yeah, it's very, and then why many of these cases are less severe, think about it this way. If you're playing a tournament or basketball game, do you want to go into it cold without having practice at all, or knowing you're having any tactics? Or do you want to have something ahead of time, or be rehearsed or practiced? Or if you're taking a test, do you want to study ahead of time, or do you want to take it cold? So vaccines are your cell's ways of studying how to combat against a pathogen ahead of time rather than trying to deal with it. Yeah, think of it this way. Would you rather take a test cold, or would you rather take a test you're studied for? So this is what vaccines are there. They're kind of there to help your cells build up these immune defenses ahead of time instead of trying to do it on the fly. So it's like that saying, if you're ready, you ain't got to get ready. That's how vaccines work. Okay, so how do you go from this to this? So how do you go from a fertilized egg and then become a full human? So how do you start off one cell and then end up as a fully formed adult? Well, so, and not only that, but how do we also do this? Ooh, look at this kind of like gnarly wound. But what happened to that wound after? Notice that it healed up. So the thing is that how do we also replace lost cells? Are these two separate questions? Well, there are two separate questions, but they have a common answer. So are you able to do that? Cell division. So this is how you're able to make more cells in your body and also replace lost cells. And this is how you do it. So what we have here is mitosis and meiosis. But mitosis is how you're able to generate multiple cells having the same DNA as the original cell. So mitosis and meiosis, and these are terms I definitely want you to know. So parent cell, a cell about to make two new cells. And when you have a parent cell, it makes two daughter cells. And notice that these daughter cells, compared to the original parent cells, they have the same genetic information. And human cells do not have only 6 chromosomes. They're just showing it for the sake of ease of presentation and keeping things from being very chaotic with 46 of these in one image. But this is a key take-home point. So with the parent cell here, and when it undergoes mitosis, you have two daughter cells that are identical. So they have the same amount of chromosomes, and if everything was normal, then they have the same DNA sequence. So if nothing went wrong with mitosis and there's no mutations, you will have the same DNA in all daughter cells. Now what we have over here with meiosis. So meiosis, tomato, tomato. But what we have here with parent cell is dividing, but now you have 4 daughter cells. And notice that in this original parent cells, each of these daughter cells have half the chromosomes as that original parent. So that's one key difference between meiosis and mitosis, is that you have in mitosis, you should generate the same, pretty much virtually identical in the nucleus cell every single time you do mitosis. But in meiosis, what you have is a halving of that chromosome. So what we have here is spermatogenesis, and then so for the male reproductive system, from one spermatocyte you generate a spermatogonium. You generate 4 sperm with this half amount of chromosomes as the original spermatogonium, and in females you have the same thing happening, but instead of so here's a key difference. So what we have with mitosis, mitosis, you're resulting in 2 identical, so they have the same amount of chromosomes, same genetic sequence. But in meiosis, you have 4 sex cells, and they have half the amount of chromosomes as a parent. And why is that? Well, when you're fertilizing a new egg, what do? You actually take those 2 halves, one from the male, one from the female, one sperm and ovum, and then you combine those halves to form a new full set. Yeah, so can daughter cells become their own new parent cells? Yeah, or yep. So daughter cells, if they start to replicate, then they become new parent cells. So it's like, where do you start? Where do you end? So this is how you're able to generate. Your cells divide, and this is how you're able to go from one cell to a fully developed human like all of you are. And let's see, cellular cleavage always visible on the telophase, cellular cleavage. So it depends on so if you look at this cytokinesis, I think that's what you're talking about. So yeah, with cytokinesis you do have forms, it's whether it's visible, I think that's a little too much detail than you need to know at this point. But yeah, cytokinesis is referring to how these cells finally pinch off and go from one parent cell and become 2 daughter cells. Okay, so how do the cells divide? Well, they do mitosis, but mitosis is only one part of how a cell's life cycle. So what happens is that there's multiple phases. Now the cell cycle, so the first phase is cell growth, or the G1 phase. Then you have your synthesis phase. And what happens during the synthesis? So G1, what do I want you to know at this point? G1 is where your cells are getting ready to divide. So they're kind of building up nutrients, they're making more organelles, they're making sure that all the resources are there for them to actually divide. And then they enter S phase. So S phase refers to synthesis. And what are they synthesizing? Well, the thing is that every time you're in a cell, remember, if you're talking about mitosis, you want to go from one cell and generate 2 cells with the same DNA as the original parent cell. So what you actually need to do is actually double the DNA so that you can produce double the cells, right? So how does it do that? Well, it actually takes the original DNA in your nucleus, or in the nucleus of a cell that's about to divide, and then it unravels it, and then it makes it into single strands, and from each single strand it uses base pairing to generate 2 new strands. I'm showing with this yellow backbone over here. But what do you notice about this? Well, it started out as this, but now with those new strands through synthesis, look at these 2 strands. They have the exact same sequence. So this is why base pairing is very important. It preserves information not only in going from transcription, but also in generating a new identical DNA molecule from one original molecule. So again, base pairing is very important in preserving it, and this is why if there's problems with base pairing, that can alter the information in a DNA molecule or even RNA. So why is S phase necessary? Well, you have 23 pairs of chromosomes. So if you've ever done those genetic tests like 23andMe, that's what the 23 is referring to. And there are 46 total. Why? Because you actually inherit 23 from your father, biological father, and 23 from your biological mother. So this is why you have 46 in each human somatic cell. And if your DNA didn't duplicate, what would happen? Well, pretend like this is your, or like say you have information from your friend is giving you their notes, lecture notes. What if instead of photocopying it, they took this and then halved it and gave you half? And what if and then your other friend wants some of your notes, and then instead of having it and photocopying it, you did another half. And you kept on doing half and half and half. What do we have? We have a reduction in the actual amount we're getting, right? So this is why with DNA you need to duplicate it, because if you didn't synthesize new DNA every time, this is what would happen to your genome. Every time you divide it into half, you would split those 3 billion and reduce it by half every single time. So it would dwindle down to nothing. If you know calculus, what happens if you take a fraction and do it to, or actually I think that this is a math class, don't worry about that. All right, so then synthesis, the main goal of synthesis phase is to double your DNA so that you're able to have an equal amount of DNA that's identical to the original parent cell when you actually divide. Then there's a G2 phase. Do I want you to know the difference between G1 and G2 in specifics? Not at this stage. Just know it's the growth stage that happens after S phase. G1 is before S phase. G2 is after S phase. And this is between that and mitosis. So mitosis, now this is what happens after G2. And say these are the stages of mitosis. This picture is pretty much all I want you to know for this class. I think maybe Intro to Biology or Cell Molecular Biology might want you to know more about this. So this cleavage furrow and where these things, like prophase is kind of like you're preparing, metaphase is when it meets in the middle. Anaphase, well, this is where it breaks apart. Telophase is where it starts to pull this like at opposite ends, and then cytokinesis is when you finally cleave something. But actually cytokinesis starts kind of around this part right here. So what do I want you to know? Well, especially with those centrioles, this is why the centrioles they're not actual like membrane bound organelles, they don't have membranes, but they're very important kind of helping to align and pull these chromosomes and distribute them so you have an even amount of chromosomes between each cells. So that's what we have. So I think of it this way. I like to think that centrioles are kind of like little fishing poles. They kind of throw their lines to the chromosomes, and they're able to kind of pull them and reel them into opposite sides of the original cell to each daughter cell. Your cat likes walking on a keyboard during class. Oh, that's funny. Yeah, I know. It's like, I know, I know that like that happened with Mr. Peanut during a meeting too. Yeah, I know all about pets interfering with class, but I mean I can't get mad at him. And plus he was like I know he was actually behaving last time. But okay, so this is for your reference, but in terms of what I actually want you to know, this part up here, but this part down here is FYI if you're taking other classes as well. So these are stages of mitosis. You should definitely know, like this is the classic I mean, so many qualifying exams at the basic level use this. So these are the stages. One of my favorite mnemonics for this is I'll pass my anatomy test. So I love mnemonics that just don't tell you like the first letter. They actually have an order to it, multiple layers of information. So these are the stages of mitosis in order. Now, common mistake is that yes, mitosis is very complex, but a common misconception is that mitosis is a majority of a cell cycle. But if you actually look at this image over here, it's like 8 or more hours in G1, 6 to 8 hours in an S phase, 2 to 5 hours. Mitosis, even though it looks like 25% of the cell cycle, it's actually the shortest part of the cell cycle. So it's the fastest part, a lot happens, it's very important, but it's not the longest phase despite being very complex and important to your body. Okay, so this is the cell cycle. So G1, then S, where you double and duplicate your DNA, and then G2, and then you have the mitosis, and then now you have 2 new daughter cells. There's also G0 or G0, G0 phase, which is quiescence, or especially if you're talking about like cancer cells and cell division, is senescence. Senescence is actually a little more complex than that, but yeah, this is what G0. So it's kind of like if a cell is in the regular cell cycle, it's dividing constantly, but your cells can kind of be idle and be put in neutral. So they're still alive, but they're not dividing. So when they're alive but not dividing, this is what we call the G0 phase. So they could divide, but they're choosing not to. So this is the quiescence, kind of like idling and staying in place. So still active, metabolically active, but not actively dividing. But the thing is that cells can exit G0 phase and end up in the cell cycle again, but that means they're actually going to divide. Now, why is this important? Well, the thing is that now we know how to generate more cells, but how does your body get rid of cells it doesn't need? And this is through cells that are damaged. So sometimes your cells get damaged, which is a natural part of life, and sometimes your body during the process of building itself, it's like, we actually don't need this tissue. Or say like if you're like me and then you've taken an absence from the gym, your body is like, oh shoot, you haven't used these muscle cells. Let's get rid of them because you don't need to burn all these calories. So this process is called programmed cell or actually one of these ways your body can get rid of cells it doesn't need is this process called apoptosis. And what happens here is that notice that these cells over here, notice that they're kind of shrinking and dying off. So apoptosis, that's what's happening over here. These cells are going from normal cells, but they're dying off, and this is actually controlled. It's not just dying off at random. There's machinery in your cells that actually triggers this process. Now, why would you need this kind of process? I mean, does it make sense? You might think, why do I want my cells to die off? Well, when you're developing, you actually generate extra tissue, and this is what happens if you don't. So this is a natural process. If you have web fingers, don't worry about it, but most of us, like during regular and most normal development, we lose the webbing between our fingers. Like this is why some animals have webs between their fingers, because they don't undergo, the cells over here, they don't undergo apoptosis, but ours typically do. So maybe some people might have more additional tissue between that. So sometimes this is your body's way of getting rid of tissues it doesn't need. And the thing about apoptosis, this is another way your body gets rid of cells that are too damaged, and they might eventually replicate out of control. So what can cause cancer? Well, genetic predisposition, that means it's in your genes. Like that's about 5 to 10% of cases. But the majority of cases are due to some sort of mutations in your DNA. Now the cool thing about your cells is that they have a lot of different genes that help to kind of check the integrity of your cell's DNA and say like, hey, this cell is too damaged. Maybe we should get rid of the cell and before it has a chance to become cancerous. So the thing is that, remember, nucleic acids contain information, and what can mutations do to the information in DNA? Well, if you have a mutation in DNA, it can cause a change in mRNA. If you change the sequence of mRNA, that can change the translation and the sequence of proteins. So that's what we see with like DNA mutations. Sometimes they can cause a protein to be abnormal, or a protein that's essential to terminate before it's actually fully made. So basically it's causing some sort of alteration. And this is what can happen in cancer. So this is advanced. Don't worry about this. This is the reason why I didn't include it. But the thing is that genomics instability and mutations can cause cells to misbehave because, again, all the information and all the potential things your cells can do is encoded in your DNA. But say a DNA you have some sort of mutation that instead of a cell saying like, okay, I'm only going to grow this much and respect the borders of this tissue, it can start to divide out of control and start to invade surrounding tissues and grow to a size beyond its normal limits. So typically, if a cell gets too damaged, it undergoes apoptosis, but this is why often cancer, you have these cells that somehow turn off those other genes that help to initiate the process of apoptosis. That's why cancer develops. I know it's a little advanced, but just letting you, if you're really interested in cancer, that's what happens. Or one thing, the major drivers of cancer. And one other thing that can cause mutations is radiation. So if you have something like, one common way of causing mutations in DNA is through ultraviolet exposure. So ultraviolet light is a form of radiation. Actually, visible light is radiation. And radiation, even though it's not going to give you like burns like gamma rays, but it does have energy. So yeah, and microwaves have energy too. That's how you heat up your food. So pretty much like, or if you've just been out in the sun or just under any sort of light, infrared lights, those have energy. That's why they have those infrared, like if you've been to a restaurant where they have like the lights shining onto a food to keep it warm, that also works. Ultraviolet actually has more energy than visible light. So what it can do is actually break chemical bonds. And when it does that, it can cause mutations in DNA. So when that happens, what happens? Well, if your DNA is mutated, that mutates mRNA, that mutates proteins. So gamma rays, they have even more information, and they don't just break bonds. Sometimes they can break whole molecules altogether. So this is this is why with radiation, like if you ever had an X-ray, that's why they put on those lead vests, especially if you're trying to get like a leg X-ray. They put it over your reproductive organs because you don't want that happening to your cells that are responsible for making your ova or sperm. But the cool thing is like knowing that cells need DNA to replicate. The other side of it is that chemotherapy works this way. Now, when we're talking about general chemotherapy for cancer cells in general, cancer cells divide very quickly. So what's one way you can kind of like screw around with the way they divide is to actually throw chemicals that actually alter DNA and actually, so again, when a cell divides, it actually includes the enzyme that duplicates DNA is called DNA polymerase. But this is a classic cancer drug. Don't worry about memorizing the molecular structure. But it's kind of like if you have a polymerase coming along and then encounters this little molecule in the way, it's kind of like throwing a wrench in the tracks. It's going to kind of cause that enzyme to halt, preventing it from synthesizing a new DNA molecule. And there's also things like these nucleotides. Here's uracil, here's a variant of that. So this is one example of how chemotherapy works. It kind of screws up with like different chemotherapies for general chemotherapy. It tries to mess around with the cell cycle and different parts of the cell cycle. Ooh, good question. Why does chemo cause hair loss? So the thing is that when you lose a hair, something has to replace it, right? So your hair cells are always generating new follicles. Thing with chemotherapy, if you're interfering with the reproduction of cancer cells, you're also interfering with the reproduction of your hair cells, but not also your hair cells, other cells that are regenerating as well. So that's why chemotherapy, the thing is that it's a very kind of a blunt, or how should I put it? Cancer cells divide faster than your normal cells, so they would uptake these chemotherapeutic agents as well, but there is collateral damage because you just don't have the cancer cells. You also have your normal cells as well. So this is why you often have people who have cancer and they're undergoing chemotherapy, they have all these side effects like the nausea. That also affects the lining of your GI tract. I mean, that also affects wound healing and your immune system. Your immune system is always generating new cells. That's why people who, especially in